It's a great honor to be invited to give this talk. In preparing for it, I realized that I've had a long relationship with this meeting. I was at the inaugural meeting, I think in 2000 in Miami, and this is my fourth meeting where I've given a talk. And so it's, I want to underscore Brian's comments, extraordinarily important that we meet you guys and interact with you. It really transforms the way we think about the research that we're doing, and it's also a humbling experience. And I wanted to start out by saying a few more words about Pierluigi, who, as Brian mentioned, actually Pierluigi mentioned, is not only a valued colleague, but also has become a close friend over the years. And in preparing for this, I was fortunate to have access to his autobiography, which he shared with me during its production phase. This is now published in the Journal of Neuropathology and Experimental Neurology, and I encourage you to look at it. It's a great bedtime reading. This is Pierluigi as a youngster in Imola, where he was born in Italy during the Second World War. He trained at Bologna University in the medical school there. He graduated magna cum laude, and he became interested in research as a resident working with Professor Lugaresi in the Department of Neurology and Psychiatry at the same university. He came to the U.S. in 1966 to study at Harvard University, and that's where he met his wife Lucy. And he then, in several months later, joined the lab of Nick Ganatis at Penn to kind of train in EM technology. He ended up staying in the U.S. and becoming a U.S. citizen to a great good fortune, and eventually transitioned to Case Western Reserve University, where he became the director of neuropathology. It's really important, I think, to understand the contributions that Pierluigi has made over the years. He in collaboration with Dr. Lugaresi, really identified fatal familial insomnia as an inherited disorder, in fact, an inherited prion disease in a number of seminal, really important papers in the New England Journal of Medicine. And also went on to show in a landmark paper published in Science that the polymorphic variation was caused by genetic variation in the gene for the prion protein. And this was not only published in Science, but also featured prominently in the magazine Discover. He showed in collaboration, this is a really an honor for me to have been collaborating with him on this and other papers over the years, but this one in particular, showing how prion strains work, how prions really encode information. And I think it's probably best to use Pierluigi's words to describe the importance of this study. So to quote Pierluigi, we decided to collaborate on the transmission of FFI with Stanley Prusner, who seemed to have the best transgenic mouse models expressing human PRP. Piero Paci, a talented and productive postdoc from Bologna, and I couldn't believe our eyes. Transgenic mice inoculated with familial CJD200 or FFI brain homogenates displayed type 1 or type 2, mimicking those of the type 1 and type 2 in the patients, and that's shown here. These differences in the migration patterns were reproduced from the patient into the transgenic mice. This finding showed that human PRP scrapie types were reproducible despite the different mouse versus human brain microenvironments. Stan quickly observed that type 1 and type 2 reproductions also occurred in the absence of the pathogenic mutation of FFI or the codon 200 mutation, which the transgenic mice that we made didn't carry. Therefore, the inoculated type 1 and type 2 types carried all the information required to produce themselves in the transgenic mice with no apparent nucleic acid intervention. In my opinion, the results of this study, published in 1996, significantly strengthened the protein-only or prion hypothesis, for which Stan deservedly received the 1997 Nobel Prize. Other highlights of Pierluigi's career include a really seminal, important classification of CJD types that was published in the Annals of Neurology. In addition to describing FFI, Pierluigi also described a novel human prion disease called variably protease-sensitive prionopathy, which is a newly described sporadic disease. In 1997, he established the National Prion Disease Pathology Surveillance Center at Case Western Reserve under the aegis of the Centers for Disease Control and Prevention, and he had a nearly 19-year tenure in that directorship, and this was an extremely important time. It occurred during the time that variant CJD was a major issue. Pierluigi remains active in the field. This is a recently published study in Brain in 2024, last year, with Pierluigi as the senior author, and it really studies an aspect of prion biology dear to my heart, and the role of post-translational modification. And so it's remarkable to see him still active in the field. As we've heard, Pierluigi had a really close relationship with the CJD Foundation. Again, to quote him, the CJD Foundation was moved to Akron and very successfully expanded by the foundation president, Florence Kranitz, creating the conditions for a very productive collaboration with the National Prion Disease Pathology Surveillance Center. One of Florence's most important initiatives was the annual CJD Foundation Family Conference. 3 days spent with patients' families was a special time, moving, and very enlightening. It gave me a glimpse of the other side of human prion diseases and of the profound sorrow and pain that they cause. I believe that this experience gave me a better sense of my work. I encourage physicians and biomedical researchers not in direct contact with patients and their families to seek out similar experiences. So this is Florence, Neil Cashman, and Pierluigi at one of the conferences over the years. And I want to finish by thanking Pierluigi for his valued collegiality and also his friendship. This is a photograph in his apartment in Manhattan some years ago, and more recently in Santa Fe, New Mexico. We were involved in a program project grant for many, many years, and so it was great to see him on an annual basis, and I really miss that interaction. Okay, so switching gears a little bit. I want to talk to you. I want to give you really an overview of kind of the things that we do in the lab and the kind of ideas that we think about. You know, we're very used to getting into sort of micro detail when we describe our results, and I want to avoid doing that today. So I hope this is valuable for you, and it gives you a flavor of the kind of things that we do and the kind of ideas that we think about. I want to begin by acknowledging the other researchers in the Prion Research Center. This is, in addition to CASE and several other centers internationally, this is a really important international center for studying prion disease research. I think also it's important for me to cast the human prion diseases that we're really interested in today into a wider context, and that is these are diseases not only of human beings but also of animals, and there's an important interaction between the animal and human prion diseases, not just clinically, but also we can learn so much from studying diseases in animals that can be applicable to studies of human prion diseases. So, of course, these diseases include scrapie in sheep and goats, mad cow disease or bovine spongiform encephalopathy, or more recently occurring disease called chronic wasting disease. Any hunters will know about this. This is a major epidemic in North America and now more recently in Europe. And of course, these are the human prion diseases, and they include variant CJD, Kuru, sporadic CJD. And these are, as we know, fatal transmissible neurodegenerative disorders that frequently occur as epidemics. Chronic wasting disease is a major epidemic, bovine spongiform encephalopathy likewise. They epitomize an unprecedented mechanism of protein-mediated information transfer. It was thought for many, many years that these diseases were caused by viruses, and I'll get into this, and I think it's important to understand that and really set this in context. The capacity for prions to transmit between species is really unpredictable. So this is how prion pathology looks, spongiform degeneration, reactive astrocytic gliosis in the brain, and in some but not all cases of prion disease, the deposition of amyloid plaques in the brain. So there are many features over the years, and these diseases have been studied for many decades now, that suggested a viral etiology. They were transmissible, they had rapidly progressive clinical phase, very similar clinical presentations. The agent was filterable and titratable, and importantly, it exhibits strain properties. So we all remember COVID and how different strains and variants were emerging. It's not too distant phenomenon to what we know about in prion diseases. Experimental transmission goes back to these studies in the early 20th century from these French workers, Queel and Schell, who showed that scrapie could be transmitted from the brains of diseased sheep into normal animals. Chronic wasting disease, as I've mentioned, is a major epidemic, and it's a highly contagious prion disease of cervids, and these include deer, elk, moose, and other cervids. And it's also spread from North America to South Korea. This is the same disease. But this in these Nordic countries in recent years, it's also been established. The strain properties of the agent in North America are very different from these emergent forms in these Nordic countries. Just to talk for a minute or two about BSE, mad cow disease. This was a major epidemic. This is foodborne transmission of prions caused by contaminated feed that was fed to cattle. The epidemic peaked in 1993 with 1,000 new cases a week. This is a remarkable, for those of you who remember, a remarkable phenomenon. There were something in the region of 180,000 cattle diagnosed with BSE in Great Britain, and it's thought that several million were actually infected and entered the food chain. And of course, we now know that by virtue of entering the food chain, it caused a novel human prion disease called variant CJD. So we know that these diseases have zoonotic potential. Fortunately, even though this was tragic for the people that were affected, these are predominantly young adults and teenagers and families who were also affected by this. The numbers were not as large as we had initially thought they might be. Other human prion diseases include iatrogenic CJD, and these are caused by accidental medical procedures. Kuru was an extraordinary disease of people in Papua New Guinea, and this was caused by ritualistic cannibalism. And this was the first Nobel Prize in the field that went to Carlton Gadusek for really discovering that this was actually, it wasn't known as a prion disease at the time, but a transmissible neurodegenerative disease. And this is the work of Clarence Gibbs in collaboration with Carlton Gadusek. And Bill Hadlow, who was a veterinary pathologist, realized that the pathology in these Kuru-affected patients, when he saw it, was very, very similar to scrapie. He knew that scrapie was transmissible, as I've mentioned, and so suggested that this might also be a transmissible disease. And in fact, that was the case. They were able to show transmission to chimpanzees and other non-human primates. You can imagine that this is extraordinarily ethically contentious, extremely expensive, and so an alternative approach was to try and use normal laboratory rodents, rats, mice, hamsters, and so on, to study transmission of prion disease. And indeed, this was a very important breakthrough, and I'll be talking about this for the rest of the talk. The problem is that like other viral diseases, other infectious diseases, I would say, there's a barrier to transmission between species, and this is called the species barrier. So this is exemplified here. Scrapie, when we try and infect, this is a kind of a thought experiment here, when we try and infect normal mice with a preparation of scrapie for an infected sheep brain, it's a very inefficient process. It takes any animals that do get sick only do so after a very long incubation time. And most animals don't get sick. However, you can take that material, retransmit it back into the same genetic host, in other words, mice, and this now becomes a relatively efficient process. So this is an example of how we think about the species barrier, how these prions can adapt across a species barrier. The other important aspect of these studies that were largely done in Edinburgh in the 1960s by Dickinson and his colleagues was that it became clear that there were different strains of agent. And in the prion world, we define strain properties by the time it takes for the animals to develop disease, the incubation time, and the pathology that these animals have in their brains. And these properties are stably maintained. It's remarkably reproducible. And this is an extremely, it was a puzzling phenomenon initially. How can this occur in the absence of nucleic acids? So there were some unusual features of the infectious agent that were inconsistent with a viral infection. There's an complete absence of any immune response. The agent is extremely resistant to inactivation. As I've mentioned, very long incubation times, and there's no detectable nucleic acid. An example of long incubation times is the study by John Collinge and Mike Alpers showing that it could take up to 50 years between the actual exposure in these kuru cannibalistic feasts and the onset of disease. So very, very long incubation times, extremely unusual. And this is the work of Tikva Alper, a radiation biologist working in the Hammersmith Hospital in the 1960s, who showed that unlike other infectious agents like bacteria and viruses that could be inactivated by UV light treatment, that wasn't true of Scrapie. And this was the first evidence that there was no nucleic acid involved in this infectious agent. So Stan Prusner was determined to find the infectious agent. Instead of finding viral particles, he found these assemblages of amyloid protein. And to summarize an extremely long and tedious study, a series of work over the years, he elaborated his hypothesis, and the idea is that this is a disease of protein conformational change. We all express this protein, and during disease, the normal shape of the protein changes in ways that we don't fully understand, but we're trying to uncover the mechanism of how this occurs. So PRBC is the normal form, PRB scrapie is the disease-associated form. PRBC and Scrapie have different biochemical properties that we'll be hearing about, I'm sure, later today. One is differential protease resistance, and this has been a really important hallmark of how to study these disorders. So the idea here is that PRB scrapie during disease imposes its abnormal conformation on the normal form of the protein, resulting in the exponential accumulation of additional PRB scrapie. And this exponential rise in infectivity was reminiscent of how viruses work. And this is why, really, for many, many years, the idea was that these were viral diseases. So this hypothesis was not met with open arms. It was highly contentious at the time, and it, in one way, because it contradicted a major tenet of biology, and that is the central dogma, what we call the central dogma, in that information flows from DNA through RNA into protein. The idea that somehow proteins could bypass that and have their own means of transmitting information was actually heretical. So our research goals are to understand the mechanism of prion propagation, the molecular basis of how strains work, how strains encipher heritable and mutable information in the absence of DNA or RNA. And we want to understand the factors controlling how prions evolve, because they clearly do, and adapt in response to various selective pressures. And most importantly, the zoonotic risk of emergent prion strains, and in particular chronic wasting disease. So going back, 30 years, this is the seminal study that I was fortunate again to be involved with when I was a postdoc at Stan Prusner's lab. These were the second transgenic mice that were made that eliminated the species barrier. This builds on the work of Mike Scott, who showed that the same was true by expressing hamster PRP. Here, we generated mice expressing the human prion protein, and we showed that we could eliminate the normal barrier to transmission of CJD prions into these mice, into wild-type mice, by expressing the human prion protein. And what this told us was that the basis of the species barrier was the constitution of the amino acid sequence of the protein. In other words, the differences in amino acids between the mouse and the human protein were the basis for why mice were resistant and why these mice expressing human prion protein were susceptible. We had a tractable model which for the first time we could study prion diseases without having to resort to non-human primates. And this was really a major breakthrough in the field. And it also allowed us to kind of refine our ideas about how replication occurred. And the idea here is that PRP scrapie, shown in squares here, interacts with the normal form of PRPC, and by virtue of this interaction induces a conformational change in the normal protein. So this countdown, I'm seeing 18 minutes. It means I've got 18 minutes left, right? Okay, good. Right. Just don't want to bore you to death. So how do prions encode heritable strain information in the absence of nucleic acids? Prion strain properties are inherited on passage from one generation to the next. That much is clear. How does this occur in the absence of DNA or RNA? And so getting back to this study, this really breakthrough seminal study where we collaborated with Pierluigi on this, really showed that this information is somehow enciphered within the shape of the protein. And we take this for granted. Well, those of us working in the field take this for granted now, but this was really a breakthrough notion at the time and a very important study. And this has recently been validated using an approach called cryo-electron microscopy, where Simon Manker and Byron Coe and others showed that different strains of the prion protein, these amyloid structures, had subtly different shapes that were linked to their different strain properties. So really this idea has been now validated at the molecular level for the first time. So we were also, in addition to generating transgenic mice where we could for the first time study human prion diseases, transmission of CJD in particular, we also wanted to see if we could model inherited human prion diseases. As we all know, these occur at an incidence of about 10 to 20% of all human prion diseases. They include familial CJD, GSS, and FFI, as I've mentioned. And they're caused by autosomal dominant mutations in the coding sequence of the gene for the prion protein. And these can either be octopeptide inserts here or point mutations, and this is out of date. There are many other mutations now that have been described. So this was actually work initially of Karen Chow and now Karen Ash, and was followed up by me and as a postdoc and also in my lab. These mice expressing the mutant form of GSS with the mutation at codon 102, it's codon 101 in mouse, spontaneously, without inoculation, develop disease. And these are control mice that express high levels of the wild-type protein without that mutation. Those mice never get disease. So now we have a model that recapitulates spontaneous disease. It recapitulates the cardinal features of the pathology in the brain here, these amyloid plaques that I hope you can see. And also it recapitulates the biochemical features. This is a study that was done recently by my graduate student, Joe DeFranco, who's here in the audience, showing that we can now really study the biochemistry of this mutant protein conveniently using this mouse model. Okay. So over the years, we made, and I'm not going to bore you with the details, made and published many, many studies based on this idea that by expressing prion protein genes from different species, that we could render those mice susceptible to prions in those species. So for example, in addition to making humanized mice, we also made mice expressing the sheep prion protein that was susceptible to scrapie, cattle prion protein, they were susceptible to BSE, and so on. And we were also able to model at-risk species, including horses. We asked if horses might conceptually be at risk in the same way that sheep might be at risk to exposure to BSE prions. We were also able to model the effects of polymorphic variation, which we know is important. And so this was a major thrust of our research over the years. However, I mean, there are major drawbacks. Even though these models are extremely valuable, there are some significant drawbacks associated with their use and their development. One is that, and this is how these mice are made, that we microinject DNA into the nucleus of one of the two nuclei in a fertilized embryo, and these genes then integrate randomly into the chromosome of the developing mouse. Okay. What this means is that it's very difficult, in fact, it's impossible to control the copy number, how these genes integrate, where they integrate, how many numbers of these genes integrate. And consequently, it's very difficult, if not impossible, to control the levels of expression of those proteins. This is important because overexpression can lead to, even though it can be useful in some aspects, it's not always the most useful phenomenon to deal with. Different expression vectors are used by different groups, also making it difficult to compare the results of different transgenic mouse models. It's challenging to assess the effects of heterozygosity, which is an important phenomenon, important genetic phenomenon. And most importantly, I'd like to convince you, for many diseases, even though we know that most of the disease outcomes are a result of expression of this disease in the central nervous system, it's very important for some diseases, including chronic wasting disease, that peripheral expression, there are events that occur in the periphery of an infected animal, which are really, really important to disease pathogenesis. And it was very difficult, if not impossible, to recapitulate all of those aspects using these transgenic mouse models. So we decided to take another approach, and this was based on our studies of chronic wasting disease, which have really garnered most of our attention over the recent decades, since I've been in Colorado in particular. Deer and elk have identical prion protein gene sequences, and they differ only at one residue T26. Deer encode glutamine and elk encode glutamate at this position. And for reasons I won't go into, we wanted to understand the effects of differences, amino acid differences at that position. And the most precise way of doing that was to change our engineering strategy. And instead of random integration of these transgenes, we decided to have a more targeted approach where we targeted the actual gene of the prion protein in the mouse and replaced the coding sequence with either elk or deer. And this allowed us to precisely study the effects of that single amino acid residue. Okay, now this approach didn't come without some risk, because previous studies had shown that this strategy was not a very efficient way of eliminating the species barrier. It was thought that you needed some level of overexpression in order to get disease. So these mice were made, they expressed normal wild-type levels of the prion protein. Instead of mouse, now they encode either elk or deer PRP. And what we found, to our surprise, was that these mice were susceptible with reasonably rapid incubation times, so really allaying those fears that they wouldn't be susceptible. The mice expressing E at 226 had faster kinetics, faster disease onset than these mice expressing Q at 226. And we were able to ascribe this difference because of the way that we engineered these mice so precisely to that single amino acid difference at 226. And we also saw effects on pathology. So this is a very, very precise means of now studying chronic wasting disease that we're now applying to other diseases, including human prion protein diseases. So using these mice, without going into the details of this, we published several different studies now where we were able to compare the strain properties of North American CWD with these newly emergent forms that are appearing in Norway, Sweden, and Finland. And we found that the strain properties of those newly emergent forms in these Nordic countries are completely different to what we know to be the case in North America. So getting back to this notion of the importance of peripheral pathogenesis. Okay, so this relates to the highly contagious transmissible features of chronic wasting disease. We think it's because The prion protein replicates not only in the brain but also in the periphery of these animals. This is what helps it be so contagious amongst animals, and it's now spread to all of these states and provinces in North America, and it's uncontrollable because it's the only disease that we know about in wild animals, and it's highly concerning. So what I'm showing you here is that these transgenic mice that overexpress the deer prion protein, whereas they're highly susceptible and have rapid incubation times when we inoculate directly into the brain, that's not the case when we inoculate into the periphery, intraperitoneally. These animals take a very, very long time, even though they overexpress the prion protein. And moreover, they fail to accumulate prions in the spleen, in the periphery. And this is very different from these knock-in, the gene-targeted mice that I just described to you. Those mice, when inoculated either directly into the brain or peripherally, the incubation times are no different. So this is a major difference between the precise knock-in models and the overexpressing transgenic mouse models. And the reason is because these mice, unlike the transgenics, accumulate and replicate prions in the periphery as well as the brain. So they recapitulate the natural cardinal features of chronic wasting disease that the transgenic mouse models don't. And I want to mention this study because this is another landmark study, and again, this is the work of Joe DeFranco, who's the lead author on this paper, just published in the Proceedings of the National Academy of Sciences. We showed that the strain properties of the infectious agent that we got out of these diseased mice differed depending on the route of inoculation. In other words, we got a different strain when we inoculated directly into the brain that was different from when we inoculated intraperitoneally, peripherally. What this tells us is that it's really important to have mouse models that accurately recapitulate the normal features of pathogenesis using this knock-in approach as opposed to a transgenic approach. And it also tells us that the strains can adapt and evolve in ways that we didn't really understand before. So based on those results, we decided to go back to the drawing board and remake knock-in human mice, gene-targeted humanized mice expressing the human prion protein sequence instead of deer and elk. Now we're using the human coding sequence, and we're modeling this extremely important change at codon 129, encoding either methionine or valine, that I'm sure all of you know about. So the results in our gene-targeted deer and elk mice indicated that targeted physiological expression of PRP from the normal mouse prion protein locus provides an improved platform to assess strain diversity and the role of peripheral compartments in pathogenesis. By extension, we realized, we hypothesized actually, that these findings suggested that gene-targeted mice offer a more refined approach to addressing the zoonotic potential of animal prion diseases. In other words, a model for assessing the risk that humans have to exposure to animal prions such as chronic wasting disease. And we know that this is a problem. Mad cow disease has transmitted to humans in the form of variant CJD. So we created these newly created knock-in mouse models, these gene-targeted mice expressing M129 and V129 human prion protein. And again, this was a very important collaboration between my group and that of Pierluigi at Case Western Reserve. Case Western provided 6 sporadic CJD cases for transmission studies into these mice. And for comparison, we also inoculated these overexpressing transgenic mice. And these mice were inoculated either directly into the brain or peripherally into the peritoneal cavity, right? And this is what we found. These are transmissions of a so-called MM1 type 1 sporadic CJD into these mice. There's 3 different isolates, 1, 4, and 6. And you can see here that the methionine MM1 isolates transmit more readily, more rapidly into the mice expressing M at 129, much more delayed disease into the mice expressing V at 129, and intermediate incubation times into mice expressing the heterozygote. In other words, the normal genotype of people with M and V at 129. This is summarized here for all three isolates. The V isolates were different. They only transmitted into the V knock-in mice, not into the M or the heterozygous mice. And these are the results in the overexpressing transgenic mice. So now we have a very precise model to understand the impact, the transmissibility of these sporadic CJD cases without the complications of overexpression. And we're able to very precisely understand the effects of M and V differences at 129. So showing you here the accumulation of abnormal prion protein in the brains of these patients, recapitulating these differences, these type differences, the so-called type 1 and type 2 differences. Okay, so I told you that these deer and elk gene-targeted mice were susceptible to peripheral inoculation, and this is shown here. What about these humanized transgenic mice? Could we also induce disease by inoculating them into the peritoneal cavity instead of directly into the brain? The answer is no, not very efficiently. So this is a major difference to our findings with chronic wasting disease. These sporadic CJD infectious preparations did not induce disease efficiently, with one or two exceptions, but generally speaking, in any genotype, with very few exceptions, as I mentioned. So again, a big difference in the properties of the infectious agent causing CWD compared to the sporadic CJD prion properties. And that's just reiterating that point here. So I'm going to close now, and I want to acknowledge the grant funding, which I hope we still have, but we don't know right now. It's a very uncertain time, as I'm sure you're all aware. But several, I've already mentioned the program project grant. This is a long-standing grant, collaboration between Pierluigi, myself, Claudio Soto, other members of the Prion Research Center at Colorado State, including Jason Bartz and Candace Mathiasen. And I want to acknowledge this training grant that supported Joe DeFranco, partly supported Joe DeFranco's work over the years. And Joe is about to defend his thesis, and God willing, everything will go well. And, I have no concerns about that. Other members, Alyssa Block, Carlos, two very talented postdocs, Dana Lowe, Jitong Shi, are other graduate students in my lab, and Hannah Sehun and Jenna are really important technicians in the group without whose help we wouldn't be able to do what we do. And I won't mention all these previous contributors. Jifen Bian, I will mention because he made major seminal contributions to a lot of these studies. The late Ed Hoover, Candace, Amanda Woman, Mark Zabel, Jason Bartz, Claudio Soto, Pierluigi Gambetti, Sylvie Benestad, and Jason Bartz. Well, I've mentioned him twice, but yeah. So thank you very much for your attention.